Hey guys, welcome back. In this session, we are discussing last 20-year immunology questions which came in GATE Biotechnology paper. So let's get started. Question number 41 says, energy refers to, it is inability of T cells to produce or respond to antigen. So option C, unresponsiveness to antigens is the right answer. Question number 42 says, based on the heavy chain, which one of the following antibodies has multiple subtypes? So only IgG and IgA has subtypes. IgG has 4 subtypes, gamma 1, gamma 2, gamma 3 and gamma 4. So option D, IgG is the right option. Question number 43 says, which one of the following complement proteins is the initiator of the membrane attack complex? So four main complement proteins are the initiator of membrane attack complex, that is C5b, C6, C7 and C8. So option D, C5b is the right option. Question number 44 says, production of monoclonal antibodies by hybridoma technology requires. So cells are taken from the spleen of the mouse. So option A, splenocytes is the right option. Question number 45 says, ABO blood group antigens in humans are differentiated from each other on the basis of, so they are generally differentiated from each other on the basis of oligosaccharides present. So option D, glycoproteins is the right option. Question number 46 is, match the cells in group 1 with their corresponding entries in group 2. So mast cells provide defense against helminthic infection. Dendritic cells contain long membranous projections. Neutrophils contain eosinophilic granules, and natural killer cells expression of CD56. So option B, that is P4, Q2, R3, S6 is the right option. Question number 47 is, match the antibiotics in group 1 with their modes of action in group 2. So chloramphenicol inhibits protein synthesis by acting on 50S ribosomal subunit. Rifamycin interferes with RNA polymerase activity. Tetracycline inhibits protein synthesis by acting on 30S ribosomal subunit. Quinolone interferes with DNA replication by inhibiting DNA gyrase. So option B, that is P3, Q4, R1, S2 is the right option. Question number 48 is, junctional diversity of antibody molecules results from. So generation of junctional diversity starts as the proteins recombination activating gene 1 and 2, RAG1 and RAG2, along with the DNA repair protein responsible for single-stranded cleavage of the hairpin loop and addition of series of palindromic P nucleotides. Subsequent to this, the enzyme terminal deoxynucleotidal transferase adds further random N nucleotides. The newly synthesized strands anneal to one another but mismatch are common. So option B, the addition of N and P nucleotide is the right answer. Question number 49 is, find the incorrect combination. So affinity maturation is a process by which THC cells activated B cells produce antibodies with increased affinity for antigen during the course of an immune response. Isotype switching refers to the generation of IgA, IgD, IgM, IgE, IgG from IgM in response to particular antigen. So option B is the incorrect combination. So option B is right. Question number 50 says, which of the following statements are correct for antigen activated effector T cells? So P is, CD4 cells make contact with macrophages and stimulate their microbicidal activity. Q is, CD4 cells make contact with B cells and stimulate them to differentiate into plasma cells. And S is, CD8 cells make contact with viruses infected cells and kill them. So option C, P, Q and S is the right option, are the correct statements.